Then our last speaker for this session is Heather Bean, who is an associate professor at Arizona State University.
Thank you, Gail, and thank you to the organizers for inviting me here. Okay. All right, so what I'd like to tell you about today is the work that we've been doing in my group on developing breath tests to diagnose valley fever and show you the results of the hard work of Emily Higgins-Kepler, who's the graduate student who's led this effort. Oh, I'm sorry. Okay, here we go. I'm on it. All right, so first I want to orient you and give you some definitions about what exhaled breath means. Exhaled breath is one of the three main waste streams of the body. We have solid waste that is eliminated through feces, liquid waste that is eliminated through urine, but we also have gaseous waste, and a bulk of that comes out through our breath. And headspace can also be considered, breath can also be considered the headspace of the blood. It is, the blood is bringing metabolites from all of our body systems to the lungs in order to get rid of that waste. And therefore, it's carrying information on the health of the lungs, but also of the entire body system. And you can perform breathing maneuvers when you're collecting breath to enrich for lung volatiles versus blood biomarkers. And so that's something to keep in mind when we want to talk about pulmonary disease versus disseminated disease. Exhaled breath, actually, when we're sampling it, it comes in two different fractions. There is the exhaled breath condensate, which is abbreviated as EBC, and it is the condensable fraction, everything that will condense out in the water portion of your breath, and that's going to include larger molecules that are human metabolites, microbial metabolites, xenobiotics, including medications. It will also contain peptides, virus particles, which is how we're sharing COVID with everybody, and epithelial cells. Cytokines will also appear in exhaled breath condensate. And then everything that doesn't condense are volatiles, the volatile organic compounds. Those are going to be small molecular weight compounds. It includes atmospheric and room air. It also includes human metabolites, microbial metabolites, and xenobiotics. My specialty is in the volatile fraction of breath. There are other groups that work on exhaled breath condensate, but the data I'll show you is about our efforts on developing a VOC-based diagnostic for valley fever. Breath-based diagnostics are making it to the clinic and to the patient. And this is a major innovation for using a diagnostic that's not based on a single specific metabolite, but more of a volatile profile of breath. In 2022, in April, the FDA issued an emergency use authorization for the first COVID-19 diagnostic breath test called the InspectIR COVID-19 breathalyzer. And so this is really exciting for our field. The FDA is working on developing the regulatory pathways that would bring a breath test to the clinic. There's also development in the realm of home use tests. And this is farther off, but it's not in the science fiction future. It's in the near future to develop home collection. The greatest advances right now are in the collection of exhaled breath condensate, where users can collect condensate at home, put it in their refrigerator or their freezer to take it to clinic. And then there are also efforts to develop real-time at-home testing, but the reality of that is a little farther out. So I want to tell you about the work that we're doing in developing a breath test for valley fever. The grounding hypothesis of this work is that when you have a different pathogen in your lung, that pathogen is producing unique metabolites that show up in your breath, but so are you. Your host immune response to that pathogen is going to differ on a pathogen by pathogen basis. And so by looking at the chemistry in the breath, we hypothesize that it will be reflective of the pathogen in the lung. The work that we've been doing has been funded through a pilot, a new investigator award from Arizona Biomedical Research Center. And I'll summarize briefly the three studies that were funded by this effort. I'll tell you about an in vitro culture analysis where we looked at the volatile metabolites produced by Coccidioides imidis and Coccidioides postdacii, and that work was published last year in MSphere. I'll also tell you about a small mouse model study, but I will emphasize that it is the only study that has ever looked at breath volatiles of a fungal infection in a mouse model, and what we're seeing there as far as the VOCs that are being produced in a valley fever infection. And then I'll show you hot off the press data of some human studies, human specimens that we have analyzed, and I want to recognize all of the collaborators on this work. So for the in vitro study, Emily worked with Bridget Barker's lab. Heather Mead was the lead scientist on this work in Bridget's lab. Heather cultured for us 12 strains of Coccidioides. There were 6 from Coccidioides postdacii, 6 from Coccidioides imidis, representing 4 major divisions of those genomes, and you can see here the isolate names that we used, including the type strains for both species. Heather cultured these under conditions that would induce spheral formation or mycelial formation, and they were cultured in triplicate. And then they sent the culture supernatants to Emily to analyze. What we observed is that the volatile profiles that are produced by these organisms are not species-specific globally, but they are very dependent on the growth phase. So in this principal components analysis, each square represents a culture that was analyzed, and the squares that are in red are postdacii, the squares that are in blue are imidis, and when you have squares, those are mycelial cultures, and the circles are spheral cultures. And we can see that we have a good intermingling of red and blue, so there's not a species-specific profile, but there is a very definitive difference between the metabolism of mycelia and spherals, and that's not surprising. These are really different life cycles. So the take home here is that we don't think that there is a lot of heterogeneity amongst the strains within species, and we don't think that there will be a need for a specific metabolite-based diagnostic for imidis and a different one for postdacii. So in the hot off the presses study of looking at human bronchoalveolar lavage fluid specimens, these were lavage specimens that were collected not for the purpose of our study, but they were biobanked at Mayo Clinic, and it represents data or samples from 55 subjects who had a lavage for some suspicion of a clinical respiratory disease. So some of these patients were ultimately classified as having Coccidioides infection, some were classified as having a bacterial infection, and those bacterial infections were quite varied. Some were classified as having a viral infection, and then some had no detectable infection. These lavage specimens, we broke them into triplicates, and if you're super jazzed about metabolomics and like complex study analyses and data analysis, come chat with me about why we do this. It helps our analysis quite a lot. But we ended up with samples that were only 200 microliters each. And then we put those into gas-type vials. We collected the volatiles from the headspace of those lavage specimens. We analyzed them by our instrument of choice, which is two-dimensional gas chromatography. And then we used random forest feature selection to identify the features that would classify Coccidioides samples and differentiate them from other sample types. And I'm only going to show you a subset of the models that have been built so far that really focus on differentiating bacterial versus cocci. So here are the infection categories that we looked at. So as of now, and we're getting some of these labels checked, we had 12 subjects and 36 samples of subjects that had no detectable infection, only 3 subjects with viral. Bacterial, it was everything. Staph, strep, but also NTMs and nocardia were represented in those. We have 17 cocci or probable cocci samples, and then we had 2 from other fungi. I believe both of them were aspergillus. So the models I'll show you will focus on the bacterial and the cocci and other fungal samples. When we used the VOCs that were detected in these specimens, and again, we performed random forests to identify the metabolic features that would differentiate the classes, and we compare in this principal components analysis the samples that were 49 cocci samples versus the 62 bacterial samples. We identified 4 VOCs that could differentiate these 2 classes with high accuracy, and the VOC numbers are just listed up here at the top. Even though the PCA doesn't look really exciting in 2 dimensions, we can see at the bottom what our classification accuracy looked like. So on the left is the label for the actual label of diagnosis, and then at the top is the predicted classification. And we can see that the classification error was about 14% for identifying those cocci positive specimens, and it was about 2% for identifying the bacterial specimens. So this is a much better classification accuracy than our current diagnostics that we're using in the lab. When we add in those additional fungal samples, we have very low representation here for those other fungus, but I wanted to show this to you. We built a separate model where we included those few other fungal samples, and we identified biomarkers to classify basically any fungal versus bacterial, although this any fungal is highly representative of cocci. And we see those same 4 biomarkers show up as we saw in the first model built only with cocci. And you can see the classification errors are very low, 6 and 2% for a fungal infection versus bacterial. So even if with a breath test, the best that we can do is to tell you that you have a fungal infection, but we don't know if it's specifically cocci, we will be making a great contribution towards clinical management and preventing people from taking unnecessary antibiotics. We have a lot more work to do in these models, so stay tuned. Hopefully we'll have an update in April. And then the last study I want to tell you about is our mouse infection study. So again, with the help of Bridget Barker's group and Marley Van Dyke and Heather Mead helped with this study and her group, they infected 6 mice with Imidis RS and 6 mice with Posidoceae strain Silvera. And we had 4 control mice that were only intranasally inoculated with phosphate buffered saline. 10 days post-infection, 2 milliliters of bronchoalveolar lavage were collected. That 2 milliliters was split. 1 ml was used to measure 26 cytokines. Another milliliter was split into triplicate samples and analyzed for VOCs. And then they also measured dissemination by doing fungal colony counts from the spleen and the brain. In this PCA, what I'm showing you is how the mouse classified based on their cytokine responses. We were a little surprised at how variable it was. But making lemonade out of lemons, what we're seeing is that if we use the cytokine responses to plot each one of these individual mice, we see that there's a gradient of immune response that is showing up on this PCA. Everywhere you see a star is disseminated disease, and if you have a darker color here, that's indicating a larger overall cytokine abundance. Over here on the far right are the 4 control mice, and we had 2 mice that were inoculated with Imidis that had very low disease responses, but we do know that it was actually different than the controls. They just did not get very highly infected. When we use the VOCs to plot these same mice onto a PCA, we can see that same gradient. The most highly infected mice that had the highest immune responses are over here on the left-hand side of the PCA, and they are being placed there based on their long volatile production. And then over on the right, there are the lower levels of infection. So what we're seeing here is that the VOCs are segregating mice by their level of immune response. And perhaps we can think about that as a proxy for their disease severity, that the VOC signature from their lung specimens is correlated with disease severity. So to summarize, in developing a breath test for valley fever, we find that coccidioides does not have a species-specific VOC profile. There's a lot of heterogeneity within species. And this indicates that, but there is overlap. There are some universal VOCs, and I didn't show you that data. So that indicates that we should be able to develop a single breath test to detect an immitis or a Possidacea infection. The VOCs differentiate cocci or perhaps just broadly fungal versus bacterial pneumonia, and we're hoping that this information will be useful to reduce improper diagnosis and antibiotic use. And from our mouse studies, we see that valley fever VOCs correlate to immune response, and we think that this may correlate to disease severity. So you would be able to use a breath test to perhaps think about whether that patient is developing severe disease and whether or not you want to go ahead and prescribe antifungals or take a wait-and-see approach. I want to acknowledge the people who did this work. Again, Emily Higgins-Kepler is the lead scientist on this study, and my collaborators at ASU, Mitch McGee and Doug Lake, my collaborator at Mayo Clinic for this work, Tom Griss, Bridget Barker's lab at NAU, Heather Mead, Marley Van Dyke, and then the funding that came from ABRC. Thank you.